So now to facilitate a Q&A session, we would like to invite our moderator for today, which is Associate Professor Christoph Winkler. Professor Christoph is an Associate Professor and Deputy Head at the Department for Biological Sciences at the National University of Singapore. He studied biology in Munich, Germany, and did his diploma thesis at the Max Planck Institute for Biochemistry in Munich. He obtained his PhD with summa cum laude from the University of Würzburg, Germany in 1994, and in 1996 he became a Human Frontier Science Program Fellow and was a postdoc at the Howard Hughes Medical Institute at University of Washington in Seattle, USA. He returned to Würzburg in 1998, where he established his own research group at the Bio Center and received his habilitation in developmental biology in 2005. So since 2007, he then became an associate professor at NUS, a member of the Center for Bioimaging Sciences, and the NUS Graduate School for Integrative Sciences and Engineering. He chaired the Department Teaching Excellence Committee and is currently the chair of the DBS Graduate Program. His research fields are in the neurogenetics and developmental biology using zebrafish and medaka as models for human diseases. So let us now welcome Professor Christoph Winkler, please.
All right. It's a great pleasure to be the moderator. I think this is a fantastic venue, fantastic location. And we heard two excellent talks. I would like to open this up for questions. How do we do this? There are microphones in the room. If you have any questions, please raise your hand and somebody will be coming to you and give you a microphone. So maybe I start with a question to John. You showed these embryos inside the placoderms, right? And you mentioned umbilical cords, meaning there's a connection between mother and embryo. On the other hand, there's also the possibility, like in guppies, of embryos having their own yolk sac, right? No link to the mother. So what's known about this link between mother and embryo?
Okay, in the first specimen of the motherfish, we found the umbilical cord, cluster of embryonic bones, and then the umbilical cord ended in a vug, a cavity in the rock filled with dark organic crystals. We think that was the remains of a yolk sac. So we think it's what's called martyrtrophy, that when you look at the umbilical cord pieces that were mineralized under the scanning electron microscope, we find very fine pores, we find what we call appendiculae, we find various structures that indicate there was probably some sort of maternal feeding as well as from a yolk sac. So another paper was published on this more recently by Kate and Zarina, and so they advocate this was martyrtrophy, a more advanced kind of viviparity. So yeah, placoderms is surprising. In the other specimens we also find bits of umbilical structure. In some of these other things we haven't published, we've got the heart, we've got the liver, we've got the alimentary canal, we've got many of the muscles preserved. So this go-go formation is like a real treasure trove of a window into the biology of these fish nearly 400 million years ago.
And in all the specimens you looked at, do you see a variety in numbers of embryos? Can you estimate what the maximum number of embryos?
Well, we only have few specimens with embryos. Most of the arthrodires have just one, but these ptyctodont placoderms have up to three. That's the only data we have on the specimens. We've only got less than half a dozen with embryos at this stage. Yeah.
There's a question there.
John, so do you think, it's very hard to say just given your last answer, but do you think all placoderms were viviparous, or do you think there was probably a diversity like there is in a lot of different sexual, different fish systems?
Well, we're keeping an open mind, but we've only got... Yeah, it's a very good question. The question was, do we think all placoderms were viviparous, giving birth to live young? We've only got 3 of the 7 families of placoderms that have embryos preserved, or evidence of internal fertilization. We still haven't found them in the antiarchs. Since we discovered sexual dimorphism in those fossils, which was only published in Nature 2 years ago, we've been scanning many of these specimens through the synchrotron and the micro-CT, looking for embryos or looking for eggs inside them, and we haven't found them yet. So I think we have a long search to go, and lots more searching for new specimens. In the old days, we used to get these specimens, put them in acid and dissolve the rock and hope that we'd reveal something. Now we've switched to a completely digital method where we can take the whole nodule unprepared, put it in the synchrotron in Grenoble, which is a very powerful particle accelerator, and then we can digitally restore the soft tissue without damaging it through the acid. So this process is far better for the result, but is more time consuming and takes more technical work to do it. Yeah.
These claspers have two ends, right? But the video you showed, only one end was used. What is the second end for?
Oh, you mean two claspers?
Yeah.
Well, one part of the anterior or the front part of the clasper attaches to the trunk shield, which internally would attach to the gonads of the male. The two sides mean, like in a shark, they've got two claspers that probably can fertilize 2 episodes of internal fertilization. That's how sharks do it. Yeah.
Any question? There's one over there, up there.
Hi, Venki, my question is for you. So you gave us 2 examples of phenotypic variation and the underlying genetic reasons. So you gave us a few different examples. So my question is that what is driving this genotypic diversity in those particular examples you gave? Or is it something unusual about the species itself? Or is it more of an accident in that sense, and we are seeing those 2 examples as the consequence?
Let me talk about loss of pelvic fin in seahorse. So pelvic fin helps in swim. It helps to maintain the balance. So in seahorse, what has happened is, also in pipefish, there's a body has elongated. Once the body elongates, there's very little use for pelvic fin. In addition, in seahorse, the entire body is covered with the bony plates. So then the pelvic fin becomes redundant. So I think this is what drove seahorse to lose its pelvic fin. So I think for each of them, probably some structures become redundant as in when they acquire some new features. And then since they become redundant, the genetic network starts degenerating. So that's how we think all this loss of phenotypic characters have come out. Whereas evolution of new one, I think it's quite complex. It's difficult to put a finger on one particular aspect and say that gave rise to, it's like, for example, brood pouch is such a complex structure. And all of a sudden you see females were carrying the young ones, and here a male is carrying. So a lot of changes have to take place. So that must have gone through stages of evolutionary changes, finally resulting in a fully formed brood pouch. In fact, if you look at other members of the Cygnathidae family, we have sea dragons, pipefish, and seahorse. In case of sea dragon, the eggs are just placed on the skin outside the body, and the incubation takes place on the surface of the body. In pipefish, they have a brood pouch, but it's not completely covered. It has just 2 flaps, and the eggs are placed inside that. Whereas in seahorse, it's completely covered, and there's a small hole through which the eggs are introduced and the young ones escape. So this is a continuum of evolving a new phenotype, and I'm sure there are a lot of factors driving these changes ecologically and genetically.
Maybe I got it wrong, but it sounded a little bit like you excluded the teleospecific whole genome duplication as the source for diversification and radiation.
I won't exclude it completely. That is still provided the additional genetic material on which the selection acted upon, and it did generate the thousands of copies of duplicate genes. But so far we have only few evidence based on these duplicate genes that have given rise to really novel phenotype. But there's no strong evidence to link this whole genome duplication to diversity of any particular group of teleost fishes. As I said, the major diversification has occurred in percomorphs, and that's happened almost 200 million years after the whole genome duplication. There was a very long time gap there.
Okay.
So immediately after the whole genome duplication, allopomorphs are there, and they are not that species rich compared to percomorph or cypriniformis and so on. So it did generate new additional genetic material, But then other factors had to come in and then utilize them and then give rise to this diversity.
And the cichlid example that you mentioned, you talked about different rates of evolution of protein coding sequences, regulatory sequences. What causes these different rates of evolution? Is it like repair
that's
mechanism?
a good question, but it's difficult to answer. You know, the mutation rate depends on, of course, several factors have been attributed to the rate of mutation. One is generation time. Shorter the generation time, higher the mutation rate. And then another factor is the metabolic rate. If the metabolic rate is higher, then you have more mutation. The body size. Larger the body, fewer mutations, and so on. But we don't know which particular factor contributed to the higher nucleotide turnover in seahorse or in cichlid fishes. We do not know that. Whereas on the other hand, when we sequenced the elephant fish, we found the DNA in this elephant fish was evolving slower than all other vertebrates. And there we could not explain why it is evolving so slow compared to all other vertebrates. So that's a very interesting question, and we need to find out more about that.
I have a question regarding the fast evolution for Venkatesh, that regarding the fast evolution in seahorse. That I just asked that what's the driving force for the fast evolution, and also how's the balance of the genome stability and instability. I think that overall it should be maintained at relatively unstable, so it can the evolution can be faster, while afterwards it has to be stabilized, otherwise they cannot survive. Yeah. So what kind of mechanism could it, and also based on what the current technologies on sequencing, also as well as those gene manipulation methods like Cas9, how far are we from to manipulate to mimic the faster evolution? So within, say, the zebrafish, is it possible that we can really randomly speed up the evolution in lab?
Okay, I think there are a few questions there. The first one was, I think I already answered when I answered Christopher. The second one, let me answer the third one, whether you can speed up nucleotide mutation rate. In experimental setup, probably you can do that. All that you have to do is knock out a DNA repair mechanism gene. But then you end up with a mutant, and it may not be fit, it may not survive. So accelerating the evolution rate is not that easy. You know, there are many factors involved in that. See, when the DNA is being copied, all the time it makes mistakes. Polymerase make mistakes. And there's a very efficient DNA repair mechanism that keeps fixing it. So it's a balance between these two. Then the selection acts. And some of the mutations are left there because it is neutral. In fact, a lot of the changes that occur in our DNA is neutral. So these things occur all the time. So whereas those which have any specific phenotypic effect, they arise very rarely, and they get selected, and that they give rise to new phenotype. So what was your middle question?
So this
or did I answer in my
I
second
think
part?
it's partly answered. And I understand. I think that because one of the evolution, for the study of the evolution, that we explain the findings, it's difficult for us to design experiment to prove those findings. Like, say, we cannot design a few mutations to repeat certain evolution from along the species. So aside from introduce those point mutations, is there any more drastic modification that we can introduce to the genome? Then
I think more than introducing mutation, probably we should be looking at closely related species that are evolving at different rates. To give an example, among mammals like primates and rodents, we are somewhat closely related. Rodent DNA is evolving 3 times faster than the primate DNA because they have a shorter generation time. So they go through more cycles of meiosis, and so they experience more of mutation accumulation. So I think this kind of comparison does give some insight, but creating mutation and testing the mutation rate, I think it takes probably millions of years to come to a balance between rate of mutation, rate of fixation, correction, and the neutral mutations that stay back, and so on, to a type of mutation. I don't think we can do that in the laboratory condition.
But your approach, testing regulatory elements in different species,
it only tells what happens after it has diverged.
Yeah.
So it doesn't tell how it has evolved faster.
There's a question there.
I'll let someone else ask a question. Sorry, I've already asked one. But you can come back to me afterwards if you want. Okay.
Hi, good evening. I would like to ask, because now with climate change and everything, the conditions, environmental conditions on Earth are changing very rapidly. So what do you think is the must-have feature that current organisms, like maybe humans or like the fish in the sea, what do you think they need to evolve to have? Or can they even do so in time to survive?
Intelligent leaders, I think, is what we need. We have the answers to climate change on a plate. We could change it tomorrow. We just lack the political will to do anything about it in some of the major countries of the world. But yeah, in terms of a major adaptation, there's a lot of work being done at the Evolutionary Biology Centre at Melbourne University about adaptations to climate change. Ari Hoffman, you probably know some of his work, where they're looking at adaptations of plants, wheat, to higher temperatures, so they're actually taking control of how some of these important food crops are going to survive increased temperatures. But it's such a complex system, climate change. It's not just it's going to get warmer or it's going to get wetter. It's such a chaotic system that we cannot predict what all the ramifications and changes are going to be. And I'm a paleontologist, I study the past, so I'd like to hear your views as well, Venti.
Yeah, I think better than asking the organism to prepare for the climate change, we should try to slow down the process of climate change. I think that's what we should be tackling it, instead of trying to modify the organisms to adjust to climate change. I think that, as you said, involves a lot of policy decisions at various levels, involvement by many people, many agencies. And that takes time. And for each kind of organism, there's a plant. For example, it's a food plant or ornamental plant or food fish or other kind of animals. I think the issues are different. I think it has to be tackled at different levels. I think the best approach is to contain it or slow down. Of course, reversing may be a big task, but at least we should try to slow it down. And first thing is people have to acknowledge that there is environmental change and it is affecting all the living creatures on the planet.
Hello. Hello, good evening. My question is, what is the path towards being a researcher in paleontology like? Was it a very tough path? And what are some real-life implications of this field of study? Thank you.
Very good question. It was a tough path to get jobs. Getting to PhD was easy, as it is in most areas of science, but getting to the next step, I had a number of postdoctoral positions. Eventually I got a job in a museum, and here I am today. But we have, it is difficult. Students have to be very competitive. They have to publish early and publish often and basically be competitive by the time they finish that PhD if they want to get to the next step. So we don't, it's like wanting to be a successful movie star in Hollywood. Some people do it, but it's hard. And yeah, I don't actively encourage students to go into paleontology unless they're really, really driven and passionate about it and are willing to work much harder than other students.
I was just going to ask another biology, which is the whole panel in general. So with the evolution of different sexual mechanisms, it seems to me that you have to evolve not only for compatibility, the morphology in the male, but also the compatible morphology in the female. And so my question to everyone is does that change the rate of sexual apparatus evolution?
I'm sure you can talk about that with your fish, but in terms of the the fossil record, all we have is external structures that we can work with and interpretation based on living forms. We know that, for example, claspers were known in placoderms since the 1930s, but people didn't believe there was internal fertilization until we found an embryo, because we don't know whether those claspers were used to transfer sperm inside a female or for some other purpose, like actually grabbing the female or something like that. So in paleontology, we're very careful about interpreting fossils until we've got good evidence that is almost irrefutable, or no one will argue that this has a different purpose or a different meaning. So your question was about the rates and change of evolutionary organs at that phase of evolution. And at all new innovations, as I said, I like to think of the human body being assembled by all these different things that were added to the model. And as the model became more and more successful, more things were added until you get the bow plan, the body plan. Now, when a new system is added, there's lots of experimentation until eventually the best one works and stays with the model. So with sexual organs, there were all sorts of bizarre clasper types in these placoderms. Multiple bones, 3 different bones in ptycnodontids, 1 large bone in arthrodires, and large fused hooked bones in these other placoderms. So there was a lot of experimentation, but we just don't have enough specimens at this stage to look at the comparable rates of evolution in them, I think. Yeah.
Yeah, I think I'll just say that I agree with you. It's the rate of change in sexual organs is slow because there is a barrier there, and unless both sexes undergo similar changes, compatible changes, it cannot be selected and it cannot be retained. So as I said, a lot of experimentation has been done. In fact, I see more diversity in the basal vertebrates, even in sharks. Elephant fish, for a long time, I didn't know they had a knob on the head. You know, it looks very odd. It has a horn-like structure on the head, and I was told that it uses it to hold on to the female while so a shark with a horn on the head. So this kind of experimentation has been done several times, and so I think it's a slow process because it's sexual reproduction and changes occur in both sexes simultaneously.
There's a question there.
Hi. Hello. Oh yeah, sorry. I just thought I'd ask a question. What do you think is the selection pressure for external fertilizations? You mentioned about internal fertilization and external fertilization being switched from 2 different strategies. So what do you think is the selection pressure for external fertilization?
Well, these are called K&R strategies. I didn't really get into it in the talk, but it goes back to island biogeography studies that you can either invest all your eggs in one basket. So K strategy is where you switch and put all your energy into rearing just a few young to a much more larger developed state, so by the time they give birth or come out of a large egg, they're almost like miniature adults ready to fend for themselves. Whereas with spawning, you're playing the numbers game. Like trout, salmon can have tens of thousands of eggs, and they hope that just a few of them make it to maturity to breed, and the rest, it doesn't matter, they're just expendable. So, you know, they're the 2 strategies. K strategy, investing in a few, R strategy, investing in many. And in different stages, or different environmental changes in fish evolution, one has had an advantage over another. So placoderms were the very first vertebrates to have a K strategy, and it worked for them. They were highly abundant, highly speciose. They were the most dominant and abundant vertebrates on the planet for 70 million years. And eventually they gave rise to the rest of the vertebrate radiation through these links to developing more complex jaws and things. I think, does that answer your question? If you look at island biogeography classic studies, that the K&R strategies were basically first looked at on populations of organisms living on small islands because... Oh, let me give you one example of modern fish. The Goodeids fish. These are fish that in marine environment, there was a few species living off North America, and then about 16 million years ago, when they invaded freshwater rivers, they went from external fertilization in water in the marine realm to switching to internal fertilization in the freshwater realm, and then speciated rapidly to about 34 species from 2 that remain in the marine realm. So there's a good example of how they switched strategies and it created also a great species radiation because they invaded a different habitat. So that's what sometimes happens with these different fish groups. Yeah.
Yeah, I think these are two alternate strategies. One, you produce few offsprings, rather than to advanced stage, so the chances of their survival are high. Whereas the other one is where you call them as broadcasters. You produce thousands of eggs and millions of sperm and release into external environment, hoping that at least a small number of them get fertilized. So they spend a lot of energy, but in these cases, you must have noticed the eggs are very small and produce in very large number, unlike internal fertilizers like shark, where the eggs are bigger, the embryos are bigger. So these are alternative strategies, and I think depending on the environment, they probably choose which is more appropriate.
Venki, I wanted to ask about the lungfish that you mentioned, the genome size, huge genome size, right? And you actually mentioned a range of sizes, or because it's an estimate, or?
No, this is based on estimating the amount of DNA per cell. That is directly correlated to the amount of genome size.
What do you think is going on? Why is the genome so big?
So one possibility is it could be a lot of junk. Other possibility is the genome could have undergone tetraploidization, duplication. But so far from what we have seen based on RNA-seq sequencing the transcript, there does not seem to be large-scale genome duplication in these fishes. So it looks like they've accumulated a lot of this so-called junk DNA in a passive manner. So as long as it is not lethal, it's not harmful, it's retained. Like something like in the human genome, nearly 98% of our DNA is non-coding. Out of that, 51% is junk DNA, which has no function. So since it was neutral, we just accumulated it. But why this fish went on to accumulate to such a large extent, it's not known. Yeah.
Being from Australia, this is about the Queensland lungfish, the genome, Neocaratidus forsteri. We have fossils of Neocaratidus in Australia going back unchanged for 130 million years, making it the oldest species of vertebrate on Earth that hasn't undergone any, as far as we can tell from the skeletal remains, any morphological sort of difference. You know, mathematically, you cannot distinguish the fossil tooth plates 130 million years old from the modern ones. So I don't know, does that have, could that affect the fact that it's a long-living, unchanged species? Would that have any effect on the genome? Or?
Coelacanth is also long-lived, and it has evolved very slowly, but it has a genome of only about 3 gigabases.
Ah, that's interesting, yeah. So it doesn't affect it, yeah.
So something interesting in lungfish fossils, so the cell size, the nucleus size is directly correlated to the amount of DNA in the cell.
Yep.
They're found in the fossil lungfish, if I remember correct, they had larger nuclei, suggesting that even the ancestral lungfish had a large genome.
And the same thing happened, well, the lungfish is at the point where fishes were about to invade land. It's the living closest ancestor to the land animals. But the same thing happened with birds. When they evolved from dinosaurs, you get a large increase in genome size measured from the cell spaces in the bone, and that was published by Chris Organ. So yeah, maybe it's because it's approaching a major evolutionary transition, perhaps.
Okay.
I just have a maybe ridiculous question. So being greedy human and intelligence at the same time, so we try to cheat the nature. So for example, when we sick, we take drugs, right? So I'm just wondering what it's your opinion of what could happen to us in the future, because or like at the moment we don't see the selection pressure that we might undergo at the moment. So we probably think that we're doing fine, but because we are cheating the nature, the rate of mutation of selecting the best fit human in this world also probably not exactly happening. So just wondering what's your thinking on this, because we might all get wiped out at one point with just one single selection because we are retaining all the weak and the strong gene at the same time.
I think you start.
Well, I'm not really studying fish and everything. It's hard to project to humans, but my general view of evolution is it's more than just mutations and the physical change of what we're going through. We humans are now in such a comfortable environment because of our techno-cultural evolution. Our brains are controlling the environment that we live in. The medical conditions of our bodies can be treated. That our evolution in future is going to be far more subtle. It's going to be less expressed in a physical way of us physically changing. It's going to be more expressed in how we manipulate our environment and come to terms with living in a changing environment through all these other ways we create and we innovate. But that's just my personal opinion.
Yeah. So I think by interfering either through medication or surgery and so on, I think we are influencing the evolutionary trajectory of humans. Just to give you an example, the head size, because of the size of the pelvic girdle, the head size was already fixed in evolution for humans. So if a child is born with a bigger skull, bigger head, either the child was aborted or the mother used to die. But then we have this cesarean operation, and doctors are able to now deliver that kind of babies. So I'm sure many of you have noticed the number of cases of cesarean has been increasing. And as a result, actually the average size of a baby's head has been increasing. This is something against evolution because of our interference, because we are able to manipulate this. We are influencing the course of evolution. So this is happening on different lines, actually.
Oh, there.
Thank you.
Sorry, it's difficult to see.
I know. I have one question about the way of reproduction. Since there's asexual reproduction and most of the vertebrates are using sexual reproduction. But we all know that in this beautiful world there are always weird exceptions, like some mythical lizard, they chose to use asexual reproduction. So my question is, how did it happen? Why did it happen? And if evolution is all about gaining advantages in competition, what is the reason behind such existence of these exceptions?
That's a very good question. I know examples of parthenogenesis is what you're talking about, virgin birth. It happens commonly in fish that are in aquaria, like sharks. It's happened as high up in the evolutionary tree as Komodo dragons. In fact, I record that in my book, Parthenogenesis, where they give birth to a female gives birth to a clone of herself. In other words, there's no sexual reproduction. It's an asexual mutation, I suppose. I really don't know what causes that. Do you know much about that, Venki? I just know it happens in the animal kingdom, but I don't know what drives it or what causes it. It's a brilliant question. I wish I could answer it for you, but I can't.
Okay.
Do we have more questions? It's really difficult to see. No? There's one.
Hi. Sorry. I just wanted to ask, I remember seeing just now that there was something about using CRISPR or Cas9 as a way to modify the genetics. Are you going to use it in any other way to modify the genome, like in a sense of research?
We are using it only in fish or other model organisms, like in worm or mouse. So we have no plans to use it in human beings or other animals. And also it's being extensively used in plant research to make changes, either to delete a gene or introduce a new element into the genome. And this is a very powerful technology. So it's actually you're quickening the process of evolution. You can introduce a new element and test whether it's viable, whether it produces more viable offsprings. So I think this is going to be very extensively used. I'm aware that it's being used at the stem cell stage in human development. People are working on stem cells. They use this to test the function of different genes or to find the genetic basis of many genetic disorders. So it is having extensive application. But I don't think anybody is going to use on animals in the wild.
I see. Okay, thank you.
All right. I think we're running out of time. I want to thank you for asking excellent questions. But I especially want to thank John and Venki for two very stimulating and good talks, right, and for a very lively discussion. So enjoy your time at the aquarium.
Well, I guess let's thank our speakers and moderator once again, right? So, ladies and gentlemen, we have actually now come to the end of the 4th series of the Chronicles of Evolution. And on behalf of the organizing committee, we would like to thank our speakers, our moderator, as well as our collaborating partner for this venue especially. Our next series will actually be organized in 2 weeks' time on the 6th of June. It will be held at NTU, Experimental Medicine Building. And we will have Francis Thackeray, who will be discussing on the human evolution in Africa, as well as Sven Pabo, who will be giving us Neanderthal perspective on the human genome. So we look forward to seeing you there. Thank you very much for this coming today.